All right, welcome to Lecture 8, Muscles and Muscle Tissue. We're continuing with our muscle mechanics, and we're going to take a look at some more in-depth facets of muscle contraction. But one of the things that we look at here is that the same principles apply to contraction of a single fiber, or also remember that that's also a cell, a fiber equals cell, and a whole muscle. And that makes sense because what happens at the fiber level is going to translate up to the muscle level. Contraction produces tension, the force exerted on the load or object to be moved. So we're going to look at some facets of tension. Can we overcome the load to be moved or not? Contraction does not always shorten a muscle. When we look at isometric contraction, there's no shortening. Muscle tension increases, but does not exceed the load. And this would be an example of if you went out onto your front patio of your house, or if you went to your car, or just something that's really heavy and tried to lift it, your muscles are going to contract, but you won't overcome that load. There's no actual shortening of the muscle. In an isotonic contraction, the muscle shortens because muscle tension exceeds the load. In isotonic, we're going to overcome the load, and we're going to shorten the muscle. The force and duration of contraction vary in response to stimuli of different frequencies and intensities. What we're saying here is that the force and duration of the contraction is going to be different depending on how much stimuli we put into it and what type of frequencies and intensities of those frequencies we put into the contraction. This brings us to a concept of the motor unit. So the motor unit then is the key structural unit that is the nerve muscle functional unit. The players in this are a motor neuron and the muscle fibers. This motor neuron that we're going to see within the spinal cord has taken its command from a primary neuron that's coming from our cerebral cortex within the motor cortex, specifically in the parietal lobe. And so if we look at a sagittal section of the brain, we have this parietal region that is the location where we have sensory and motor cortex. Within this area then, we're going to send a motor signal down to neuronal cell bodies within the spinal cord. Those neuronal cell bodies are going to send their axons off, and those axons are going to split, and they're going to terminate and branch more times at a smaller level at these individual muscle fibers or muscle cells. So the idea that we'll be looking at is the more of these axons that we recruit, the more of these neuron cell bodies then that we recruit, the different types of contractions we can have. Do we contract do we use a small amount of axonal input or neuronal input and affect a small number of fibers? Or do we fire more neurons and have more fiber input for a stronger contraction? So the relationship between these neurons and these fibers is the fundamental core of how we take a look at the differences in contraction that we have of the same muscle. And if you think about it, you can lift your bicep brachii, you can lift it in a very slow coordinated fashion without a lot of power, or you can lift it with a lot of strength. And same muscle, just more innervations of the fibers, more neuronal push coming out of that, and that's what we're going to take a look at, how that varies within these motor units. The motor unit then, we've got small motor units, and we're going to see those, for instance, in the fingers and eyes. And the concept is that for a body part or a muscle part that we have to move with more finesse, we're going to commit more neurons to that. And then in a large motor unit, these would be the motor units, say, within really large muscles in the thighs or hips. We don't have a lot of a need for the fine movement within these large motor units. So we're going to see motor units that have a different structure to them in terms of what they're controlling. So small motor units for finesse movement, large motor units for more crude, larger muscle movement. Muscle fibers, that, just like we looked at from a motor unit, are spread throughout the muscle so that a single motor unit causes weak contraction of the entire muscle. And motor units in muscle usually contract asynchronously to help prevent fatigue. You've got one motor unit firing off, and it maintains a certain amount of the contraction because it does. Each motor unit then typically has this relationship where the one single motor unit causes a weak contraction of the entire muscle. We have multiple single motor units causing weak contractions of the entire muscle. If we want to maintain a muscle contraction, we fire those off asynchronously so that we can recharge those muscle units, and that our latent periods within those contractions don't cause us to lose contractility. We fire off asynchronously, and that way we can maintain a more smooth, consistent contraction. Muscle twitch, then, is the response of a muscle to a single brief threshold stimulus. So we reach threshold and we fire that off, and we just have one brief little muscle twitch. It's the simplest contraction observable in the lab, recorded as a myogram. So myo, our muscle portion of the term, and a gram being a chart. There are three phases of a twitch. There's a latent period where events of excitation, contraction, coupling are occurring. And then there's a period of contraction where we have cross-bridge formation. Remember, in cross-bridge formation, where we're tying those myosin heads onto the actin receiving sites. And then we have a period of relaxation where the calcium is going to have re-entry into the sarcoplasmic reticulum, and then our muscle tension is going to decline to zero. As we reuptake that calcium, we reset the troponin-tropomyosin complex and block those actin binding sites. Here's just kind of a chart where we would take a look at an isometric twitch. Remember, isometric, the muscle isn't actually going to shorten. It does have a contraction. Here then, we have a single stimulus that comes in this latent period as we're, say, resetting our fibers. Then we go ahead and have a period of contraction where this is our tension over here across a variable of time. Here then, this twitch goes up to a certain level. That's where we're contracting. Then we're reuptaking calcium and relaxing, resetting all those troponin, tropomyosin heads within this portion of the region, resetting all of the troponin, tropomyosin complexes and removing all the myosin heads. When we compare muscle twitches, we can see differences in strength and duration that are due to variations in metabolic properties and enzymes between muscles. Just saying that different muscles are set up to handle twitch differently. If we take a look at an extraocular muscle, say lateral rectus is the muscle that lets you turn your eye laterally. So if you want to look at something in your peripheral vision laterally, lateral rectus does that. Very small muscle, very finely controlled. And so what we see is that muscle is going to have a much smaller type of twitch time frame than these larger muscles that are designed more for duration. When we see muscles that don't have that capability of endurance, then they're going to have a much tighter twitch response than muscles like we start getting to gastrocnemius. It has a slightly slower twitch response. Then when we get to soleus, really this major endurance muscle for the flexion of our foot, and we use that so much in walking that it's assisted by gastrocnemius, but it's the major player in this endurance activity. It has a longer range on this twitch response or on this individual muscle contraction. if we take a look at that individual twitch, then we need to take a look at how do we add those up? What does the addition of an individual muscle contraction, what kind of responses do we get? That's referred to as graded muscle responses. These are variations in the degree of muscle contraction, and they're required for proper control of skeletal movement. These responses are graded by changing the frequency of stimulation and changing the strength of the stimulus. Within this, then, we would say that a single stimulus results in a single contractile response, which we just looked at as a muscle twitch. And another look at that we have a stimulus come in, this typically like an action potential, and then we're going to have a contraction, where then we're going to have a relaxation, and that's what we just looked at in that twitch section. The response to change in stimulus of frequency, if we increase the frequency of stimulus, the muscle does not have time to completely relax between stimuli, and calcium release stimulates further contraction, and we refer to this as a temporal wave or a summation of these stimuli. And further increase in stimulus frequency we refer to as an unfused or an incomplete form of tetanus. So tetany is a extreme amount of continued frequency of stimuli. Here we see a low stimulation frequency or an unfused or incomplete tetanus. If another stimulus is applied before the muscle relaxes completely, then more tension results. This is a temporal or wave summation and results in unfused or incomplete tetanus. This is an example of you're lifting something with your muscle and you need to keep tension on it. You need to keep the muscle flexed. So we're going to keep firing stimuli so that muscle stays tensed until we're done lifting that item, and then we can relax the muscle. If stimulation are given quickly enough, we have a fused or a complete tetany. That looks like this when we have this stimuli that continues to fire, and we just have that muscle completely locked into contraction. We're fully releasing calcium at the whole time in that sarcoplasmic reticulum. The troponin and tropomyosin complexes are not covering the actin binding sites. The myosin heads are attaching to the actin, maintaining contraction, resetting through ATP, continuing to connect with the actin binding sites, and we just keep having this complete tetany or complete contraction. That's a look at frequency of inputs. Then if we take a look in a difference in the stimulus strength, then what we're taking a look at is the threshold stimulus, the stimulus strength at which the first observable muscle contraction occurs. Threshold stimulus then is sort of our baseline for where we see our first observable muscle contraction, and the muscle contracts more vigorously as stimulus strength is increased above that threshold. Contraction force is precisely controlled by recruitment, so we're pulling in more motor units, and more motor units adding in becomes a summation, which brings more and more muscle fibers into action. Previously, what we were looking at is this is a firing, say, of a similar axon. This strength then is a recruitment of more motor units. So a difference between frequency and strength. They typically go together, but they can be broken out to be looked at individually. Here's a threshold stimulus that we would take a look at. We have to hit this level in order to have a muscle contraction. And if we don't hit that, we don't have a muscle contraction. But once we hit that, then the further we go beyond that in terms of signals that we're sending through our motor unit recruitment, we're going to get to a maximal stimulus. And when we start going beyond that, there is a potential within this area where injury could occur, or we just have to be careful of how much we're doing at that point. And typically, the muscles at this point will be so engorged with blood that we start losing our capacity to have oxygen available and contract appropriately anyway. This is kind of a neat cross section if we cut the muscle, say like just in a transverse section, and you see all of these individual cells in here packed into a fascicle, and then we just start recruiting more of those cells, because each one of those is each one of these cells has a relationship with a motor unit. And so this is just showing that proportionately each one of these could symbolize a motor unit. And within that, then, the more of those motor units that we recruit, the more fibers we recruit, and the stronger the muscle contraction that we have. In response to change in stimulus strength, the size principle then says that motor units with larger neurons and larger fibers are recruited as stimulus intensity increases. Just taking a look at this idea that the muscle is innervated with all of these different motor units, and so we're going to start off with small fibers that fire off. Then we're going to start with medium fibers and then larger fibers. And so the more of these that we recruit through these motor units, the stronger our muscle contraction is going to be. That's all that we're seeing in that figure. Muscle tone, then, is a constant, slightly contracted state of all muscles due to spinal reflexes that activate groups of motor units alternately in response to input from stretch receptors in muscles. These stretch receptors in muscles are really ingenious feedback for our muscular system. They exist within all of our muscles, and this feedback is going ahead and saying, okay, we're stretched enough or we're not, and in order to maintain our muscle firmness, allowing us to be ready to respond, we're always having muscle contractions to a certain extent to maintain our muscle tone. Looking again at isotonic contractions, these are the muscle changes where we have a change in length that's going to move the load. Isotonic contractions are either concentric or eccentric. So concentric contractions, the muscle is going to shorten and does work. In eccentric contractions, the muscle contracts as it lengthens. So this is kind of an interesting scenario for you'd think, how does it contract as it lengthens? But as you think about lowering a load or releasing something, then we're contracting that muscle to maintain control over the load, but we also have those muscle fibers lengthening to a certain extent so that we can release that load. Here then, this concentric isotonic contraction, just saying that on stimulation, muscle develops enough tension or force to lift the weight. Once the resistance is overcome, the muscle shortens and the tension remains constant for the rest of the contraction. This is an idea of where when you have contraction, you overcome the load. We do this quite a lot whenever we pick anything up. And looking at this stimulus here, where we have that certain amount of tetany, where then we relax the muscle and release the weight. Within isometric contractions, the load is greater than the tension the muscle is able to develop, and so we're not going to be able to overcome the load that we're trying to lift, and tension increases to the muscle's capacity, but the muscle neither shortens nor lengthens. Just taking a look at this weight here is just too great for this muscle to overcome. So even though these fibers are contracting, trying to lift this up, it's not going to happen because the load is too great, and we never get to this threshold where we are able to overcome that. That's one of the differences within isometric contraction. The energy that we get for contraction is through ATP. It's the only source used directly for contractile activities. And we have available stores of ATP within the muscle that when we start using our muscle, those stores are depleted within 4 to 6 seconds. So you realize that the ATP that's stored in our muscles is just to get them up and going. It's not going to be the long-range energy that we need to keep going. But we have to regenerate ATP by the direct phosphorylation of ADP by creatine phosphate. When we think about this, we have an ADP, we have 2 phosphate units. We take a creatine phosphate, we take this phosphate unit and phosphorylate to add on here so that we get ATP. In the anaerobic pathway, when we don't have enough oxygen available, we're going to use glycolysis exclusively to do this. And in aerobic respiration, we're going to get a lot more ATP out because we have oxygen available. When we have oxygen available, we go from glycolysis into our citric acid cycle, and then we go into our electron transport chain, where we see a huge amount of ATP come out in comparison to just glycolysis. Direct phosphorylation is what we refer to as a coupled reaction, because we're going to see two different things occur. We have creatine phosphate, we have that phosphate group that we want, and adenosine diphosphate. And what we need to do then is go ahead and take that phosphate group off, add it onto the adenosine diphosphate. Out of that, we get ATP, and we get creatine. One of the things that tests that we can take a look at is how much creatine the kidneys are clearing, to see maybe if we have kidney issues, because a lot of proteins should not be clearing the kidneys if they're healthy. And creatine is one of the tests that we can take a look at to assess kidney function. There's no oxygen use, so this is one of our anaerobic processes. We're going to produce 1 ATP per creatine phosphate, and we're also going to create creatine. And the duration of this energy provision is about 15 seconds of energy. So you can imagine how much this has to happen to maintain a muscle contraction or workout for any amount of time. In the anaerobic pathway then, at 70% of maximum contractile activity, what happens is the muscles start to bulge and compress blood vessels. What that does is impair oxygen delivery. And so out of glycolysis, we produce pyruvic acid, which in the presence of oxygen would move on into the citric acid cycle. But without the presence of oxygen, that is going to transfer into sort of a storage version of pyruvic acid, which is lactic acid. Lactic acid then is going to diffuse into the bloodstream. It can then be converted back and used as fuel by the liver, kidneys, and heart. Then it's converted back into pyruvic acid by the liver. We can see that our main energy source is glucose. We have a lot of energy within that, with our C6H12O6. There's a lot of energy that is within this, and what we do then is, by breaking that glucose down in glycolysis, we push off a certain amount of ATP, and we also push off pyruvic acid, which, like I was saying, if there's O2, it's going to enter the citric acid cycle, and if there isn't, then it gets stored as lactic acid. The duration of this energy provision is around 60 seconds or maybe a little bit more. Once our muscles start being engorged with blood and we can't get oxygen into them, we start having a harder time providing the energy that we need to keep them going. In the aerobic pathway, this is going to be about 95% of the ATP during rest that we need, and during light to moderate exercise. So when we're not engorging our muscles with blood, but we are still exercising, we can use the aerobic pathway because we have plenty of access to oxygen. But we're going to burn stored glycogen, typically stored within the liver. Then we're going to also burn the blood-borne glucose, like through our food. We'll burn pyruvic acid from glycolysis and any free fatty acids that are available to fuel our muscle needs. Here then, just looking at that, glucose, say from glycogen breakdown or delivered from the blood, to pyruvic acid into the mitochondria, and we also send in fatty acids, amino acids, to do a similar thing as pyruvic acid. And out of that, then, instead of 2 ATP that we see in glycolysis, we get 32 ATP from the end result of this entire process. The one thing that this isn't really showing, this is just saying that both the citric acid cycle and the electron transport chain are happening in here, and that's what's providing us with this greater amount of ATP. This then can give us energy for hours. When oxygen is present, we can really run for a lot more amount of time. Our muscles can function a lot better, and we can go do what we need to do. So this is kind of a cool chart out of your book, just saying that at 6 seconds, we're burning the ATP that's stored in the muscles. At 10 seconds, we're forming ATP from creatine phosphate and ADP. At 30 to 40 seconds to the end of exercise, The glycogen that's stored in muscles is broken down to glucose, which is oxidized to generate ATP. We see glycogen as a storage version of a chain of glucose molecules that we take a look at within muscles and the liver. Then at prolonged duration exercise, we see that ATP is generated by the breakdown of several nutrient energy fuels by aerobic pathway. And that's why if you get on a treadmill and you go for fat burn rather than cardio, you'll notice that your fat burn program on the treadmill is going to give you more moderate exercise so that you can plug in those fatty acids to provide fuel for your muscles. And in many ways, you would think, oh, the harder cardiac workout would do what I need to burn fat, but that's not necessarily true because you can move into that anaerobic state and not access those fatty acids. So muscle fatigue then is the physiological inability to contract. And this is going to occur when we have ionic imbalances of either, say, potassium, calcium, or phosphate. This is going to interfere with our coupling mechanism. Within this, then, prolonged exercise can also damage the sarcoplasmic reticulum. And of course, that's going to interfere with our calcium regulation and release. If we don't have the container, or if we damage it, we're going to have problems. A total lack of ATP occurs rarely during states of continuous contraction and causes contractures or continuous contractions. When we don't have ATP, we can't recock those myosin heads, and they're going to stay stuck to the actin, and we have a problem within that. Extra oxygen needed after exercise for replenishment of oxygen reserves, glycogen stores, ATP, and CP reserves. We always need to reset these systems so we can keep functioning on the next movement that we need to make. And we're also going to have conversion of lactic acid back to pyruvic acid, and then we can go ahead and get into our glucose realm. And glycogen then is just those stored quantities of glucose. So glycogen is just a lot of rings of tied together glucose. We have a lot of heat production during muscle activity. It's estimated that about 40% of the energy released in muscle activity is useful for work, but around 60% of the remaining energy is given off as heat. We can have dangerous heat levels if we don't radiate that heat off the skin and sweat. Sometimes when people exercise, like in sweatsuits, they can have serious problems, essentially cook their internal organs, specifically the brain, because they're not able to evaporate that sweat and cool themselves.